Today we're talking about chlorophyll. We are going to start with the structure and characteristics of chlorophyll. This is a light-harvesting pigment in photosynthetic plants, algae, and bacteria. It is found in the chloroplast of plant cells. It reflects green light and absorbs red and blue light, red being 600 to 700 nanometers, blue being 400 to 500 nanometers. These are lipid-soluble and non-polar. They are also weak, non-covalent bonds, so they are easily extracted. They are tetrapyral with a magnesium complex in the middle. There are 7 chlorophylls in nature. A and B are found in plants, and these are present 3 to 1, A to B. They only differ on the carbon 3. B is more stable because of the withdrawing effect of the formyl group on the carbon 3. Health benefits of chlorophyll are decreasing the size and incidence of tumors and also protecting from aflatoxin exposure. Ways that chlorophyll can be affected, one of those is acid. At a lower pH, there are hydrogen ions more capable and present to replace the magnesium. This will lead to pheophytin, which is more olive green in color. At a higher pH, the methyl and phytyl esters are removed. This creates chlorophyllin, which is a brighter green. With heat, the chlorophyll is bound to lipoproteins and protect it from acid, but when the heat is lowered, that protection is gone. Hydrogen ions are also more permeable across the cell membrane, making them more available to replace magnesium. Also, heat will cause proteins to coagulate and release the magnesium, which will in turn be replaced by hydrogen ions and make pheophytin. If you continue heating the pheophytin, it will lead to pyropheophytin. Then there's enzymes, which is chlorophyllase is the enzyme that affects chlorophyll. It removes the phytyl group from chlorophyll and makes it soluble in water. This makes chlorophyllide, which is more stable than chlorophyll, or pheophorbyde, which is when the magnesium has been removed. This enzyme is active in plants between 60 and 82.2 degrees Celsius, which is when they're harvested. They are blanched to inactivate the enzyme. If you do not blanch and completely inactivate it, it will be active even during freezing. This will lead to chlorophyllin, which is an even brighter green. And then there's light and oxygen. Chlorophylls are connected or protected by carotenoids and lipids. So this prevents them from degradation of heat because they are light sensitive. But when this protection is destroyed, you get an irreversible bleaching or oxidizing. And then there's the metallocomplex, which is when zinc and copper replace magnesium in the chlorophyll. And they will do this more readily. This is more stable in acid and will increase the green color. Food processors will add zinc to blanching water to cause this reaction and make their vegetables more green. Copper, though, is not permitted in the U.S. So, we're going to do a quick review, starting with chlorophyll. As I said, you remove the magnesium. This is because of acid. You're going to get pheophytin. If the phytyl group is removed because of the enzyme, you're going to get chlorophyllide. If you do not inactivate this enzyme completely, you will get chlorophyllin. And if the pheophytin is continued to be heated, you will get pyropheophytin. Now, if the magnesium is removed from chlorophyllide, or the phytyl group is removed from pheophytin, that gives you pheophytin. 4-bide. And whatever is left to be removed, magnesium, CH3 minus, or phytol, that will leave you with pyrothio 4-bide, which will be gray in color and ugly. And that's all.